Hello, and welcome to our Genes and Diseases and Symptoms presentation on Alzheimer's. In this presentation, Rebecca, Juliana, Kelly, Kathy, and I will focus on a GO2R dataset about hippocampus transcriptional response in patients with Alzheimer's. So what is Alzheimer's? Simply put, Alzheimer's is a progressive neurodegenerative disease and the most common form of dementia. The most common and well-known symptoms include memory loss, as well as the loss of cognition and the ability to perform simple tasks that come with the disease's progression. Though the cause is not completely clear, scientists say that it seems to be a mix of genetic, lifestyle, and environmental factors. One clear feature of Alzheimer's is the plaques and tangles that develop in the brain. Plaques form between nerve cells, while tangles are knots of brain cells. Another feature would be the loss of connection between neurons in the brain, making us less able or unable to communicate between different parts. Finally, Alzheimer's disease affects over 5 million Americans as of today, and its complications make it the sixth leading cause of death in the U.S.
In our analysis, we used GSE-1297 incipient Alzheimer's disease microarray correlation analyses. This study performed microarray on samples from the hippocampus, a region of the brain known to be associated with Alzheimer's. There were 31 samples, including 22 from patients with varying degrees of Alzheimer's and 9 controls. The researchers at the University of Kentucky were specifically looking for differences in hippocampal transcriptional response between the control and Alzheimer's patients, based on their microarray data.
So after considering the given data on hippocampal genes, we decided to separate the subjects into 2 groups instead of 3. Initially, the subjects were separated into control, moderate, and incipient, which means that their Alzheimer's was just beginning to emerge. We decided to group them into 2 groups, control and incipient/moderate, since these groups already started showing signs of Alzheimer's and will potentially have genes that are correlated with Alzheimer's. The questions that we developed are: what Alzheimer's markers can be found in the expression of specifically hippocampal genes? And are there any pathways that are affected in these patients that already have Alzheimer's? And what genes are significant in that pathway?
So, after we analyzed our dataset through GEO2R and string, we found that there were many genes associated with the long-term potentiation pathway shown here. The circled proteins are ones linked to specific genes in our dataset. Some of those genes are listed here. The most significant gene was CREB-BP, which is linked to the CBP protein. This gene had a log FC value of 1.0316, which meant it was expressed more in our Alzheimer's group than the control. The linked CBP protein plays a role in activating DNA transcription and is known to be a part of Krebs signaling, which converts short-term memories into long-term ones. A potential gene mutation in this protein could affect its function and therefore long-term potentiation.
So the idea behind the long-term potentiation pathway is that synapses get stronger over time based on recent activity in the brain. And this is suspected to be instrumental in the mechanisms of learning and memory. So changes to that pathway could affect memory, which is generally a common indicator of Alzheimer's disease. So differences in expressions of genes in this long-term potentiation pathway could lead to phenotypic traits that are associated with Alzheimer's. In the future, we would prove causation through further trials that specifically measure patients with differences in the pathway with all other variables randomized. If it is indeed a factor causing Alzheimer's, we would develop drugs that target genes in this pathway to try and alleviate some of the symptoms. And we would also perform trials in mice using gene editing to find out how changing different genes in the pathway affects Alzheimer's symptoms.